Well guys, welcome to Learner's Big Book. In this new presentation, we are going to discuss the experiment where we are going to calorimetrically measure the total phosphorus or the phytic acid present in foods and animal feeds. Now here these are the slides which we have prepared, which are about 16 slides, and here we have tried to detail the every required information for the experiment and why we are doing this estimation. The calorimetric method of measuring the total phosphorus and phytic acid in the foods, consuming food and the animal feeds. Now in the next slide, here we are going to discuss the term anti-nutrient. Anti-nutrient term is one of the research topic. Here they are researching on a few of the anti-nutrients which are present in the form of these phytic acid, phenolic compounds, enzyme inhibitors, saponins, lectins and hemagglutinin. So what they are and how they are present? They are present in the natural or even in the synthetic form. Now here what they are going to do, food is going to have the nutrients. Those nutrients has to get absorbed, but these anti-nutrients are going to interfere with those nutrients and it is going to avoid those nutrients to get absorbed. Now how they are found? They are found in the foods of variety of the compounds. So now here they are never toxic. They are not necessarily toxic, or they are never toxic, and the nutritional value where it is going to lead to the deplete in the essential amount of the nutrients to be get available within the gut of the animal, where because of this it is going to lead to the indigestible of that particular consumed food by the humans or the animals. So here what are those? When you are going to see, you are going to get astonished, because each day we are going to consume these particular foods. So this is our daily routine where we used to consume the soybeans, whole grains, chickpeas, beans, lentils, nuts, spinach, broccoli, peppers, eggplants, tomatoes, chocolate, tea, cocoa and all this. So make a big list of the anti-nutrients. Now here these are the 4 anti-nutrients where they are going to become the groups or the kinds, where they are going to have their specific features of avoiding or making the essential nutrient to be unavailable or going to lead to indigestible within the animal gut or human gut. Because of that, the nutrient is going to get retained within the food which is getting digested, but the breaking of that particular nutrient is not going to take place and not going to get absorbed. Here, as we have discussed, that we are going to estimate the phosphorus and phytic acid present in the foods. So here, out of these all anti-nutrients kinds, we are going to discuss the phytic acid. In the next slide, we are going to discuss the phytate. It is the phytate, or it is also called as the phytic acid, and other names are like this: the inositol hexakis phosphate, and generally it is abbreviated as INSP6-IP6. Here again the other name is that 1,2,3,4,5,6 hexakis dihydrogen phosphate myo-inositol, and the IUPAC name is this one, that is the cyclohexane 1,2,3,4,5,6 hexyl hexakis, where dihydrogen phosphate is present. So here first we understand the chemical structure. Chemically it is a 6-fold dihydrogen phosphate ester of myo-inositol or cis-trans hexol. These are the names, and here the structure has been said like this will be the myo-inositol, where there is no phosphate bonded. Here you can see all the OH groups are present. Here only the inositol is present. Now here, when you are going to say 6-fold dihydrogen phosphate ester, that is going to be present in the IP6. Here this will be the inositol core, which will be surrounded with the 6-fold dihydrogen phosphate. Here each H2O3PO is the dihydrogen phosphate has been bonded with the myo-inositol. So here this is going to form inositol hexacase phosphate IP6. Here this is how the phytate or the phytic acid is present, or the structure is seen. Now, because of the structural, chemical and physical properties, phytates can be found in many biological systems, including most of plant and mammalian cells. Now here it is going to be the main part of all the mammalian cells and also the plant cells. Here you can observe that phytic acid or the phytate, behalf of being an anti-nutrient, it is also a essential component to the biological system. So here in the further slides we will study how it is the anti-nutrient, but you have to browse and understand how this phytate or phytic acid is the essential component. So here, when you are going to consider further, these phytates also present in the form of the phytins, where they are going to be present in the form of calcium, magnesium, potassium mixed salts, where they are going to be present in the huge amounts in the plant tissues. So this is the structure of the phytate or phytic acid. Now in the next slide, in this slide we are discussing regarding the monogastric or non-ruminant animals and the human digestive system, where these are digestive systems which doesn't have the phytate degrading enzymes endogenously. So because of that, what will happen, this particular phytate is not broken down in the digestive system of monogastric or non-ruminant animals and even in the human digestive system. So here, when you are going to compare this phytic acid is the one of the main component which is going to act as the principal storage for the phosphorus. As the topic of this experiment, or the title of this experiment itself, is the estimation of the phosphorus and the phytic acid in the food or the animal feed. Now here the phosphorus is going to be present in the principal storage, that is the phytic acid. So in this structure we have already spoken that inositol phosphatase, that is the INSP6, is going to have the phosphorus attached to it. Now here this is the structure which is going to, this myo-inositol or the inositol core is going to have the dihydrogen phosphates attached to them, and here it is going to become the huge core for the phosphorus to be present within them. So these phytates, they are present in the plant tissues. So the plant tissues are going to have the phytate, where it is going to have both the myo-inositol or the inositol and the phosphorus together. Now here this phosphorus bound to inositol predominantly is going to constitute to the whole sample of that particular cereals, legumes, nuts and oils, seeds to be 60 to 80% of the total phosphorus. So whatever the sample you are taking, if you are going to take the sample of 100 grams and if you are going to get some amount of the phosphorus identified, okay, within that amount of the phosphorus present in that sample, 60 to 80% of the total phosphorus from the inositol bounded phosphorus. Now here, and also in lower concentration, it will be present in root, tubers, fruits and berries. So the high concentrations will be present in cereals, legumes, nuts and oil seeds, and in the lower concentration it will be present here in this. Now if further is that the high phytate concentrations will be especially seen in the bran and seeds. So here you can see bran is used for the animal feed and seeds also used for the animal feeds, and even the seeds we are consuming, that is human beings. So here there is the high concentration of this particular phytate. Because of that there will be phosphorus concentration which will also be high. Now here further, the phosphorus is one which is going to make the phytate an anti-nutrient. First is that it cannot be broken, so it is going to remain intact in its structure. So because of it is going to be in its structure, whatever the phosphorus is present in its. Now here what will happen, importantly, it is going to have negative effect on the essential dietary minerals, where the phosphorus within the phytate is going to chelate with the transition metals. That is, it is going to make the bond with the transition metals. It is going to grab the transition metals into its fold, like the calcium, magnesium, zinc, ferrous, copper, manganese, to form insoluble complexes that are not absorbed. So here already we have spoken, phytate is not already broken down. Because of that it is not absorbed in the digestive system. After that it is going to form the complex structures with the calcium, magnesium, zinc, ferrous, copper, manganese. It is going to hold these particular metal ions within it, and because of this remain within that food itself, and it is going to get excreted or going to come out of the body of the human being or the animals. Monogastric animals consuming any food, in that food there will be other nutrients also, like proteins and multivalent ions or the metal ions, and minerals are going to present. So within that food itself the phytate is also going to present. Now here, depending upon the amount of the phytate present in that food and the phosphorus is available within that particular phytate, that is going to correspond to the reducing the nutrients bioavailability to the gastrointestinal tract. Now here it is going to contribute to the deficiency of the all the metals which are required by the biological system. But here further, when you are going to consider these phytates again have the effect of indigesting or avoiding the dietary proteins to be get absorbed by the digestive system, or it can also inhibit digestive enzymes to show their action on the food. Here how it is going to happen, it is during the extreme pH. The extreme pH can be acidic also, it can be basic also. At the basic extreme pH or acidic extreme pH, the phytates will be having their effect more on forming the indigestible complexes with dietary proteins, and also it is going to inhibit digestive system from showing the action on the food. So when the going to make complexes with dietary proteins, definitely the digestive enzymes will not going to have any action to make it to or break it to make available to the digestive system to get absorbed. Now this is what the point here. Further is ruminants. As we discussed here, monogastric or non-ruminants. Here it is the ruminants. These are the animals which are going to have rumen microorganisms which are going to produce the phytase enzyme. This phytase enzyme is lacking here in the non-ruminant and human digestive systems. Now here, when you're going to see the structure, here you can see phosphorus is reacting or chelating with the transition metal ion, zinc, here iron, calcium, like this. They are going to make the complex structures where it is going to avoid these particular metal ions to be get absorbed by the digestive system. Now here further, how this particular phytate can be degraded. It can be degraded by enzymatic method or non-enzymatic means. Now here, enzymatic is nothing but hydrolysis, generally occurs during biological processing of food or feed. Now here you are going to break down that particular phytate with the help of the enzyme, and you are going to make that food or feed so non-ruminants and human beings where they can consume and absorb that particular phytate and phosphorus, and it is also going to lead to the absorption of that particular transition metals or the essential dietary nutrients. Now here further, this enzymatic degradation is a more selective and isomer specific. Here very specific to the isomer in the sense a particular enzyme is going to act on a particular kind of the isomer of the component to be get degraded. So here it is very selective in its nature. Now further is non-enzymatic hydrolysis. Usually takes place when food or feed is treated with heat and pressure or with a strong acid. Now here when you are going to see, usually as we have discussed in the anti-nutrient slide, there I have given a list of food names which we are consuming daily of our life. We are consuming those foods by cooking it. That is, we are going to treat that food with heat, appropriate amount of heat and pressure. That is in the pressure cookers, or different methods will be applied during the cooking. So here it is with a strong acid. It is nothing but, again, we are using many means to make the food to get breakdown, and it is going to be treated in such a way that after consuming, definitely the food is going to be very good and nutritious. This non-enzymatic method is going to break down the phytate. Now further, here there are several methods available to determine the phytic acid concentrations in the food products. Now here we have seen many papers which are having many different modifications to these methods, where these methods, in the sense, compared to each paper there are modifications done to estimate or measure the phytic acid and the phosphorus. There is no ideal methodology to be agreed upon. Here there are many different techniques that can be used to identify the phytic acid. As I have said, the existing methodology needs to be optimized and standardized. Here the need of the optimization and standardization is very much. It is because here you can see there is no direct method that can show or envisage or give the proof that the phytic acid will be detected with the help of the specific reagent, and phytic acid's various forms cannot be detected with the specific reagent. So this is the biggest failure. Moreover, phytic acid does not have a characteristic absorption spectrum in the UV and visible light region. Now here, when you are going to see many calorimetric methods, in that you can see whatever the estimation, in the sense whatever the chemical or whatever the component we are estimating, that will be having the spectrum where we can read that particular specific reacted component and we can estimate that particular component. But here it is not so, where phytic acid lack that particular characteristic of being within the absorption spectrum, so it is not going to give any reading. Further, when you are going to consider, these are the most observed papers of the research and standard where iron phytate complex will be precipitated and further quantified or measured or calculated, McCann's and Widowson where phosphorus will be estimated, and in this calorimetrical determination of iron will be seen, and further in this the iron or inositol will be measured spectrophotometrically analysis. Here as it is shown inositol, it doesn't mean that directly it is measured. Here also again there will be the multivalent metal of the phosphorus will be present within. With the help of that the experiment will be conducted. Now further, when you are going to consider what is critical overview of the experiments whichever the paper has been presented till now, here there should be a development of the most relevant analytical principle and suppression and detection method should be applied, and there should be the overcome of the difficulties with specific chemical properties of inositol phosphates. That those chemical properties will be like interferences, absence of characteristic signals, that is the absorbance, and strong binding interactions. Now here the strong binding interaction in the sense the phosphorus which is going to react with the metals which is present in the biological molecules, that is within the same food, and it is going to make that particular thing to be in the form of the hard complexes where it cannot be further absorbed. So here this is where the experiment is showing the no promise of being one of the direct method to estimate the phytic acid, but here apart from that the experiments are good to estimate corresponding to the phosphorus present within the phytate and estimating that to estimate the phytate or the inositol. Now further in the next slide, in this it is the preparation. So here we are discussing the required materials preparation, that is the sample, assay buffers, phytase and ALP. Now here when you are going to consider, we are preparing the, that is the sample extraction where phytic acid will be extracted from samples, where we are going to take some amount of the sample, that can be any food or the feed samples, and it is going to be milled and it has to be passed to the 0.5 millimeter screen, so we can get the fine sample, where it can be done through 40 mesh ground, preferably using a ball mill, and if not you can also make your other alternative present. If you are able to ground or grind the sample very finely and you can pass that through the 0.5 millimeter screen in your lab, you can do that also, and whatever the sample you get, that has to be 1 gram after the screening, that is 0.5 millimeter screening. After passing it through that, you should get amount of 1 gram of the millet sample, and it has to be taken into the 20 ml of the hydrochloric acid, which should be 0.66 molar, and further it has to be vigorously stirred for 3 hours and left at 24 hours at the ambient temperature. So stirring should be only of 3 hours, and further it has to be left for 24 hours at ambient temperature. Further is, out of that 1 ml of the extract was transferred to the 1.5 ml of microfuge tube and centrifuged at 11,000 rpm for 10 minutes. Whatever the supernatant you get out of that 0.5 ml has to be taken into the another microfuge, which is again 1.5 ml, and there you are going to neutralize with sodium hydroxide of 0.75 molar, and the adding amount will be 0.5 ml. Now here, when you are going to consider after this neutralization of the sample, the enzymatic dephosphorylation has to be done for this sample. Now here, that will be discussed further during the procedure. Now here, when you are going to consider the other preparation, in this we are preparing alkaline phosphatase assay buffer, that is the ALP, and preparation of phytase assay buffer. Now here these two buffers are prepared to the suspension or the enzymes, that is the ALP, alkaline phosphatase is the suspension or especially enzyme itself. Phytase is also enzyme, where 80 units per ml, 12,000 units per ml is the concentrations. Or here this alkaline phosphatase assay buffer will be prepared exactly done here, that is 30 grams of glycine, which will be 400 millimolar, 13 milligrams of magnesium chloride hexahydrate 4 millimolar, 115 milligrams of the zinc sulfate heptahydrate 0.4 millimolar, and it has to be dissolved. All these three agents should be dissolved in the 900 ml distilled water, and pH has to be adjusted with the help of the NaOH pellets to 10.4 pH. The final volume has to be adjusted to 1 liter with distilled water. Now further, the weight of 27.2 grams of sodium acetate trihydrate, which is of 200 millimolar concentration, will be dissolved in 900 ml, and the pH will be adjusted to 5.5 with the concentrated hydrochloric acid. Now the final volume, again here it has to be 1 liter with distilled water. Now as I said, these are the enzymes. These has to be purchased or bought from the distributor of chemicals. So here you can get it from any good company. In the next slide, here we are preparing color or dye reagent and the standard preferences. Now here, when you are going to consider, you are going to make solution A and solution B. In the solution A, you are going to take 10 grams of ascorbic acid in 90 ml of distilled water, and has to be stirred until it is clear, and then add 5.35 ml of concentrated sulfuric acid to it, and further you have to make up the volume to 100 ml. So whenever going to use any concentrated acids like sulfuric, nitric, hydrochloric, you should add by the sides of the container slowly with the pipettes. Further, the solution B, dissolve 1.25 grams of ammonium molybdate in 20 ml distilled water. After dissolving, adjust the final volume to 25 ml with distilled water. Now here this is how ammonium molybdate is prepared. Now further, with the help of these two solutions A and B, we are going to prepare the color reagent, where you are going to take the solutions in the B:A ratio, that is 1:5 ratio. B:A solution, that is 1:5 ratio, where it will be 20 ml of the solution B mixed with 100 ml solution A. Even you can take 25 ml also. Due to its instability, you have to prepare this particular the color reagent with the help of the solutions on the day of the experiment. Otherwise, it is not going to give good results. There can be failure of the experiment also. Now here it is the preparation of working standard reference of the pure phytic acid, 30 milligram per ml, and you are going to do the phytic acid at 4 concentrations, 3.75, 7.5, 15 and 25 milligram per ml in distilled water. So the phytic acid dissolves in the distilled water, and out of this each concentration you are going to take 1 ml each concentration, and you are going to do the standard reference for the phytic acid. Now here this is the preparation of this particular working standard reference for the phytic acid. It is the principle where the principle involves acid extraction of phytic acid, which we have already discussed, where extracted the sample with the help of the 20 ml of the acid and myoinositol phosphates from food or feed sample. Now after the acid extraction, the myoinositol phosphates has to be extracted, or IP6 has to be degraded to get the phosphate separated from the myoinositol. So here the further enzymatic dephosphorylation will be done, where a specific phytase enzyme will be used on phytic acid and the lower myoinositol phosphate forms. Here whatever the IP6 which we are going to treat with phytase, it is going to get degraded to IP5, IP4, IP3, IP2, and it is going to be IP1. Here that will be because of the phytase enzyme. Now here further subsequently, that is after treating this particular phytic acid with phytase, we are going to do enzymatic dephosphorylation here by alkaline phosphatase. Now here this is going to ensure the release of the final phosphate from results, that is myoinositol monophosphate will be formed and the separation of the phosphate from the inositol is going to take place. Now here you can see, as we have discussed during the preparation slides, the use of the enzyme or the suspension phytase and the use of the alkaline phosphatase is in the principle. So these two suspensions are very important to make the change or give the effective change within the experiment. So here when you are going to consider further, you can see this is how the reaction flow chart goes, where the extracted phytic acid will be treated with phytase and the myoinositol phosphate is going to form by releasing inorganic phosphate. Now here you can see the degradation of the phytic acid will happen, where even after the degradation there will be phosphate attached to that, and here there will be again the free phosphorus in the sample. Now here alkaline phosphatase further hydrolyzes myoinositol, producing myoinositol and inorganic phosphate. Now here this is the reaction where the alkaline phosphate will be used to break the myoinositol phosphate, where the myoinositol and inorganic phosphate is going to get separated, and further the inorganic phosphate present in that particular sample, after treating the sample with the phytase and alkaline phosphatase, the inorganic phosphate which is going to form is going to be reacted with ammonium molybdate color reagent, which has been prepared and which is going to react with 12 molybdophosphoric acid. Subsequently is going to reduce under acidic condition to molybdenum blue. So the reaction is going to happen here, where phosphorus reacts with ammonium molybdate reagent by reducing it to 12 molybdophosphoric acid. So the color, and further this is going to react with sulfuric acid, ascorbic acid present in the color reagent during the preparation. So it is going to lead to the formation of the molybdenum blue. Now this is how the reaction flows. Further is, after the formation of the molybdenum blue, the color concentration of the molybdenum blue formed is proportional to the amount of the inorganic phosphate present within this sample, that is the phytic acid sample, where it will be measured at 655 nanometer absorbance calorimetrically. So this is the principle where it is exactly mentioned what the reaction is going to happen and how the reaction is going to happen and what will be the results. Now further, this is the slide where the enzymatic dephosphorylation of the samples will be done. Now here, this I have put it in this tabular form where whatever the sample you take, the sample, as we have discussed, it has to be treated with the suspensions which has mentioned in the previous slides, like the phytase and the alkaline phosphatase. Now here, when you're going to consider whichever the sample, that sample is going to have free phosphorus. Free phosphorus in the sense, before to the treatment of the sample with the phytase or the alkaline phosphatase suspensions, the free phosphorus will be present, where that is considered to be as the not bonded with the phytic acid. So if you are going to calculate or include that phosphorus during the calculation, it can give the wrong results, that is the wrong amount of the phytic acid present in the sample. So the amount of the free phosphorus is also has to be identified, and as per the principle, the total phosphorus which will be present within the sample, that also has to be identified. Here the total phosphorus of the sample is nothing but the free phosphorus which is present within the sample, and also the whatever the inorganic phosphorus which will be present with the phytic acid. That is the sum of the both the free phosphorus and the inorganic phosphorus associated with the phytic acid. So I will tell what is the need of doing this both during the tabulation. Now here, how the dephosphorylation is going to unfold. As we have taken in the extraction, during the extraction we have taken 0.5 ml of the phytic acid sample and neutralized with the sodium hydroxide of 0.75 millimolar concentration, where we add that sodium hydroxide of 0.5 ml. So after neutralizing, we are going to take only 0.05 ml out of that neutralized sample in both the microfuge tubes, 1.5 ml microfuge tube for free phosphorus and one more for total phosphorus. In both the microfuge tubes, we are going to take this particular sample extract only of 0.05 ml in each, and we are going to take distilled water of 0.62 ml here and 0.60 ml here. It should be very accurate. And further, phytase assay buffer has to be added. Phytase assay buffer, which is going to have the specific pH to make the phytase enzyme to react with the sample extract. So here phytase assay buffer has to be added of 0.2 ml in both the microfiche tubes. Further, you have to add phytase suspension, that is the phytase enzyme, to only to the total phosphorus microfiche tube, that is of 0.2 ml. And further, you are going to vortex or mix thoroughly the components of both the microfuge tubes and incubate it at 40 degrees centigrade for 10 minutes. And further, you are going to add, after 10 minutes, you are going to add distilled water 0.02 ml to only to free phosphorus microfuge tube. And further, alkaline phosphatase assay buffer will be added of 0.2 ml to both the microfiche tubes. And alkaline phosphatase suspension or the enzyme will be added only to the total phosphorus tube of 0.02 ml. So here you can see further the components has to be mixed thoroughly and further has to be incubated at 40 degrees centigrade for 15 minutes. Now further, you are going to add trichloroacetic acid, which will be of 50% weight per volume, and this will be one to stop the reaction of the components which has been added within these microfuge tubes. Especially it is going to help to stop the reaction of these particular suspensions which has been added within this particular total phosphorus tube. But we have to add the trichloroacetic acid of 0.30 ml in both the microfuge tubes. Now further, after this, both the microfuge tubes has to be separately centrifuged at 11,000 rpm for 10 minutes. Further, the supernatant has to be transferred carefully. That is, it should be of 1 ml of the supernatant probably to be transferred to the fresh 1.5 ml microfuge tubes separately, okay, for free phosphorus and total phosphorus estimation, and that is further treated with the color reagent. Next slide. Here we are going to quantify the sample by reacting with the color reagent, that is the ammonium molybdate, which we have prepared already. Now here what we are going to do, as whatever the sample we have taken from the dephosphorylation procedure, we are going to take that particular supernatant of 1 ml in the 1.5 microfuge tubes, that is both from the free phosphorus and total phosphorus microfuge tubes. We are going to take into the new microfuge tubes for the free phosphorus and the total phosphorus as labeled, and now we are going to add color reagent of 0.5 ml, and further we are going to mix it very thoroughly with the vortex mixture, and we are going to incubate in a water bath for 1 hour at 40 degree centigrade. So after incubation, the solution or the components will be mixed thoroughly again, and further approximately 1 ml was transferred to 1 centimeter path length micro cuvette. So whatever it has been mentioned here, all this has to be done accurately, because result of the calculations are dependent on this particular, whatever the use of the reagents and the amount of the reagents has been used in these particular procedures. So here next the absorbance will be measured at 655 nanometer of each solution, that is the free phosphorus and the total phosphorus of one sample will be recorded, and further the recorded absorbance values of the samples of the phosphorus standard solutions were used in the calculation of the total phosphorus and phytic acid. Now here, this is how you are going to extract the sample, and you are going to dephosphorylate it, and you are going to treat it with the coloring reagent, and further you are going to record the absorbance, that is for the single sample. In the sense, if you are going to take 5 different food samples, for each one you have to do this particular procedure, and you can estimate the absorbance of that particular samples. Now further, in this slide you can see whatever the absorbance has been recorded, that is, as we have used barley, in this the free phosphorus absorbance values were this one, and the total phosphorus is this one, and the oat this one, and the other is this one, and the wheat is this one, and the total phosphorus is this one, and the soya bean absorbance is this one for phosphorus, and here total phosphorus is this one, and the millet free phosphorus absorbance will be this one, and total phosphorus absorbance will be this one. Now here you can see, as we have already spoken, that free phosphorus is one which is already present freely, where it is not because of the phytic acid, which is where the phosphorus is bounded with it. It is already present. So this is what the free phosphorus absorbance, where the total phosphorus absorbance is 1, where the free phosphorus is also be included here in this absorbance, and also whatever the inorganic phosphorus which we have separated from the phytic acid, that is going to be present here within this absorbance. So what we are going to do, considering these two readings, we are going to subtract the free phosphorus from total phosphorus. That is, we are going to determine the amount of the exact phosphorus which is associated with the phytic acid. So whatever the subtraction has been done, that is this particular total phosphorus will be get subtracted by free phosphorus. So whatever it is, the absorbance value is present here for barley, oat, wheat, soybean and millet is the exact amount of the phosphorus which got separated from the phytic acid within these samples of 1 gram each. So that is what the phosphorus absorbance here. So this will be used further in the calculation. Before to that, we are going to discuss one more required slide, that is preparation of the standard phosphorus calibration, in the sense preparation of the calibration phosphorus, where in this the standard phosphorus will be taken as the reference for the further calculation of the sample phosphorus and further calculating the phytic acid. Now here the standard phosphorus is, we are going to prepare stock solution where 50 microgram per ml phosphorus will be prepared, where we are going to make standard solutions like labeling STD0, 1, 2, 3, 4, and these concentrations will be corresponding with the STD 1, 2, 3, 4, like 0 will be blank, 1 will be 0.5, standard 2 will be 2.5, standard 3 will be 5, and 7.5 microgram phosphorus for standard 4. It will be per liter. So you are going to prepare in per liter, and here after preparing them you can apply it for the calorimetric determination of this particular assay. In the sense, after preparing these particular standards, that is the working standards, you can collect 1 ml out of each of it and you can transfer it to 1.5 ml microfiche tubes. So each tube will be again labeled as blank STD1, STD2, STD3, STD4, standard 1, 2, 3, 4, and it will be added with the color reagent. Here there is no addition of any buffers or the distilled waters or anything. Here whatever the preparation you are doing, it is done with the help of the distilled water itself. But here after taking 1 ml each of these concentrations into microfiche tubes, there is no addition of any buffer, distilled water or the suspensions. So here it is purely directly adding of the color reagent which we have prepared of 0.5 ml in each of the tubes and vortex them, and directly you are going to do the absorbance. There is no incubation, no waiting period, nothing. So you are going to record the absorbance, then the absorbance values at 655 nanometer will be considered for the further calculation. Now in the next slide you can see here we have tabulated the absorbances, phosphorus standards as we have labeled: standard blank, standard 1, standard 2, standard 3, standard 4. Now you can see the concentrations which I have taken. After this, whatever the absorbance recorded has been seen here, it is considered to be as the absorbance 1, and one more absorbance is also recorded. In the sense, a set of standards will be prepared and absorbance will be taken or recorded, and one more set of the standards will be prepared and one more set of the absorbance will be recorded. So both will be compared to see that the experiment has given the correct absorbance corresponding to the whatever the concentration of phosphorus we have taken for the reference. So here after doing this recording of the absorbances, these both absorbances will be summed and averaged, that is added and divided by 2. So whatever the result we get here after doing that calculation, will be taken here. This one here, it is going to be standard absorbance of the phosphorus, that is after this addition and divided by 2, this will be the standard absorbance of the phosphorus. So here this particular absorbance will be considered the m, mean of the each concentration per phosphorus absorbance will be calculated. So here it is m is the mean of the each concentration microgram. This is what the concentration microgram per phosphorus absorbance will be calculated. Here how it is, it will be phosphorus concentration divided by the phosphorus absorbance will be done. So here the concentrations will be taken from here and the absorbance will be taken from here. The values of the absorbance will be considered from here and the calculation will be done and the results will be like this. So here when you are going to do, you will definitely understand, and these are also the accurate one which we have got. Now further, whatever the values we got, it will be like m standard 1 values, m standard 2, m standard 3 and m standard 4. Here all these will be for the absorbance only. It is not going to determine any form of the concentration, and it is not going to be mentioned with the microgram, that is the mass of the concentration. So here it remains to be microgram per AVS. So both is going to be mentioned here, but these are the absorbance values only. That will be m, that is the mean, and further the mean of the m will be calculated, where it will be microgram per delta A. Absorbance of the phosphorus will be taken, that is all this will be arithmetic mean will be done, and whatever the mean m we are going to get out of this will be 4.550. So this will be used in the further calculation of the phosphorus and phytic acid. Now further in the next slide, here you can see this particular graph will be drawn to understand that how good the standard phosphorus has given the reaction with the ammonium molybdate color reagent or the dye, and how it is nearly correlated, where R is the 0.9544 correlation, which has shown a perfect linear regression line. So this is to understand this one, we are going to do this particular graph. So it has given the perfect linear regression where it has given the very nearest correlation. So the reaction between the amount of the phosphorus and the molybdenum which has been taken has been reacted very closely. So here further, this is the slide where we are going to discuss the sample of phosphorus absorbance, calibration and phytic acid estimation. So here we are going to calculate both the phosphorus amount and the phytic acid amount. Now here you can see we have to consider whatever the samples we have taken and the sample weight we have taken during the initial sample preparation. We have taken 1 grams of the weight of each sample, and the extraction volume, whatever the hydrochloric acid we have taken, will be 20 ml. So further we have taken the ABS phosphorus, nothing but here this sample phosphorus after subtracting the fair phosphorus from the total phosphorus. This has to be considered here. This will be the exact phosphorus absorbance within the phytic acid, and further these values has to be subjected in this formula, that is the total phosphorus gram per 100 gram, mean m into v into f divided by 10,000 into lowercase w and lowercase v, and we are going to take absorbance of the sample phosphorus. Now here absorbance of this phosphorus will be this one, each one you are going to take during the calculation, and mean m what we have taken is the medium from the standard reference phosphorus, that is the 4.55, and V will be original sample extract volume. Original sample extract volume will be 20 ml, whatever the 1 gram of sample we are going to put in this, and we are going to further extract the phytic acid. That will be the sample volume, that will be 20 ml, and F will be the dilution factor. The dilution factor will be 55.6, that we will discuss in this later. And further, the 10,000 is the conversion from microgram per gram to gram per 100 gram. You know that the phosphorus absorbance, that is delta A phosphorus, is the absorbance change of sample. And further, lowercase w is the original weight of sample material, that is the 1 gram. That is the 1 gram here. And further, the lowercase v is the sample volume used in the calorimetric determination of phosphorus assay, that is also 1 ml. Now here, this calorimetric assay 1 ml, you know that during the absorbance recording in the cuvette we are taking 1 ml of the sample after the development of the color. And here you can see what is this dilution factor. It is nothing but, as we have taken 0.5 ml of the sample during the sample extraction, that is to neutralize it with the sodium hydroxide, where we are going to take 0.5 ml of the sodium hydroxide and neutralize the sample, phytic acid sample, and it is going to be factor of 2. Because of this neutralization, the sample of 5 ml is going to be neutralized and diluted. Again, it is diluted twice, in the sense whatever the concentration has been reduced to the half of it. So because of the addition of the 0.5 ml of the sodium hydroxide, so it has become the 2 factor. Now further, out of this we have taken 0.05 ml of the 0.05 ml of the sample. After the neutralization, we have taken 0.05 ml of the sample, and we have reacted it with 1.39 ml of the different solutions, like these buffer solutions, that is the phytase buffer, alkaline phosphatase buffer, suspensions, distilled water. So altogether it is going to become 1.39 ml. Now here, this is going to become the diluted factor of 27.8. How it will be calculated? It will be calculated where you are going to divide the 1.39 value by 0.05 value, so you will get the value of 27.8. Now here already the factor dilution will be of twice, that is the 2. It will, this particular value of 27.8 will be multiplied by 2, and further you will get the value of 55.6. so this is going to be the dilution factor. So after the subjecting these values, you will get the phosphorus amount gram per 100 gram of these, corresponding to samples and the sample weight. So in this each sample of 1 gram of barley, oat, wheat, soybean and millet, we are going to get the phosphorus amount to be of these. Now further, whatever the phosphorus amount we have got, that has to be considered to calculate the phytic acid, where it will be done with the help of this particular formula, where the phosphorus amount we got will be put on the numerator and denominator will be having a constant of 0.282. This will be because of 0.082 is the factor which will be used to convert the phosphorus content to phytic acid content, because phytic acid comprises 28.2% phosphorus. So whatever the constant value has been mentioned or the factor has been mentioned, that has to be taken or considered, and the calculation should be done. And here you are going to get these values: 2.9260, 3.7870, 1.2598, 1.4197, 1.4335. so these are the values. So here you can see that the experiment has very much clearly explained, and I hope I have not done any mistakes during the explanation. If you are going to find any mistakes or any wrong explanations, please come forward and mention in the comments, and further we will take into consideration and do the explanation related to that. And I hope I have given the much of the information required for this experiment, and I believe that you people will like this. Share this particular information which we have prepared after doing a very much hard work, and thank you very much.